Welcome to our lecture on steroidogenesis. Today, we are going to explore one of the most fascinating and vital processes in human physiology. We will uncover how your body acts like a highly efficient chemical factory, transforming a single, simple molecule into a diverse array of powerful hormones that keep you alive, help you adapt to stress, and drive human reproduction. Whether you are completely new to endocrinology or just looking to solidify your understanding, by the end of this video, you will have a clear, intuitive grasp of how this incredible biological machinery works. Let us start by imagining a massive, highly efficient car manufacturing plant. This factory received daily shipments of raw steel, which serves as the basic building block for absolutely everything they make. Depending on which specific assembly line the steel travels down, it might be hammered, welded, and painted into a compact family car, a heavy-duty pickup truck, or a sleek, high-speed sports car. In your body, steroidogenesis is exactly like this bustling factory. It takes a single raw material and, through a series of specialized steps, builds entirely different end products that serve vastly different purposes in your daily life. Now that we have our factory analogy in mind, let us look at the actual biological components. In the human body, the raw material is not steel, but a common fat molecule called cholesterol. And instead of human factory workers, your body uses special protein machines called enzymes. These enzymes act as the mechanics on the assembly line. Their job is to snip, fold, and reshape the cholesterol molecule. Depending on which specific enzymes are present in a given organ, that raw cholesterol is transformed into different powerful chemical messengers, such as stress hormones to keep you alert, or sex hormones to guide development. Building on that intuition, we can now formally define steroidogenesis. It is the complex biological and biochemical process by which the body synthesizes steroid hormones from a single precursor molecule, cholesterol. This intricate metabolic pathway does not happen everywhere. It primarily takes place in highly specialized organs: the adrenal glands, sitting on top of your kidneys, the gonads, which are the testes and ovaries, and the placenta during pregnancy. These organs are the dedicated factories, equipped with the exact right enzymes needed to carry out these life-sustaining chemical conversions. So where does the very first step of this assembly line begin? The entire process starts deep inside the mitochondria of the cell. Here, a critical enzyme called cytochrome P450 side chain cleavage enzyme, or simply desmolase, goes to work. Desmolase converts cholesterol into a molecule called pregnenolone. Pregnenolone is incredibly important because it is known as the universal precursor. This means it is the very first intermediate product from which all other steroid hormones are eventually derived. From the mitochondria, pregnenolone moves into the smooth endoplasmic reticulum, where other specific enzymes systematically add or remove chemical groups to create the final hormones. Understanding this pathway is absolutely critical because the hormones produced dictate how we survive and adapt on a daily basis. For instance, cortisol is a major product of this pathway. It is essential for managing psychological and physical stress, regulating our metabolism, and reducing dangerous inflammation. Another vital product is aldosterone, which controls our blood pressure by managing sodium and potassium levels in the kidneys, ensuring we do not faint when we stand up. Finally, the sex steroids, such as testosterone and estrogen, drive the development of reproductive organs, maintain muscle mass, and keep our bones dense and strong. Given how important these hormones are, what happens if the biological assembly line breaks down? Sometimes, a person is born with a genetic enzyme deficiency, meaning one of the workers on the assembly line is missing or defective. This leads to severe, life-threatening conditions, like congenital adrenal hyperplasia. In this condition, the body cannot produce cortisol. Because the pathway is blocked, the raw materials pile up and are shunted down a different assembly line, causing the body to overproduce other steroids. This imbalance throws the entire physiological system into chaos, highlighting just how perfectly regulated steroidogenesis must be. To truly understand how the body controls the amount of steroid hormones produced, we must look at the biochemical rate of this conversion. We can model the speed of the desmolase enzyme using the Michaelis-Menten equation. In this formula, V represents the actual velocity or rate of steroid production. Vmax stands for the maximum possible rate, when every enzyme is working at full capacity. The term S in brackets represents the concentration of the available cholesterol substrate. Finally, Km is the Michaelis constant, indicating how much substrate is needed to reach half of the maximum speed. When cholesterol concentration increases, the production rate V also increases, until it maxes out at Vmax. Conversely, if disease lowers functional enzymes, Vmax drops, dangerously decreasing hormone production. Let us apply this formula with a concrete example. Imagine an adrenal gland cell actively producing pregnenolone. We are given a maximum production rate, Vmax, of 120 nanomoles per minute. The Michaelis constant, Km, is 40 micromolar, and the current cholesterol concentration, S, is 60 micromolar. We need to find the current velocity, V. We substitute these values into our rate equation. The numerator becomes 120 multiplied by 60, giving us 7,200. The denominator is 40 plus 60, which equals 100. Dividing 7,200 by 100 gives us our final answer. Physically, this means the adrenal gland is converting cholesterol into pregnenolone at a rate of 72 nanomoles every minute. Since our cholesterol level is slightly above the Km value, it makes perfect sense that our rate is slightly above half of the maximum capacity. Once steroid hormones are produced, they travel through the blood and are eventually cleared by the liver. In physiology, a steady state is reached when the rate of hormone production exactly equals the rate of hormone clearance. This brings us to our fundamental balance equation, R equals K times M. Here, R is the rate of production, acting as the input. The variable K is the clearance rate constant, representing the drain, and M is the total mass of the hormone currently circulating in the blood. To maintain a stable hormone level, the body must perfectly balance the input R with the output, which is represented by K times M. If production increases, the mass in the blood will rise until a new balance is found. Let us see how this balance works in a patient. Suppose a patient's adrenal gland produces cortisol at a constant rate R of 15 milligrams per day. The clearance rate constant K for this patient is 0.05 per day. We want to find the steady state mass, M, of cortisol in their body. First, we rearrange our balance equation to solve for M by dividing both sides by the clearance constant K. This tells us that mass is directly proportional to production and inversely proportional to clearance. We substitute 15 for R and 0.05 for K. Dividing 15 by 0.05 gives us a final answer of 300 milligrams. This means the patient has a total pool of 300 milligrams of cortisol circulating to handle daily stress. If the liver failed and clearance decreased, this mass would dangerously increase. Now that we have calculated the rates, let us look at the entire factory floor. This diagram illustrates the complete steroidogenesis biochemical pathway. At the very top, you see our starting raw material, cholesterol, being converted into pregnenolone by the desmolase enzyme. From there, the pathway branches into 3 distinct zones. On the left, in blue, we have the mineralocorticoid pathway, leading down to aldosterone. In the center, in green, the glucocorticoid pathway produces cortisol. And on the right, in purple, the sex steroid pathway generates testosterone and estradiol. Notice how interconnected these pathways are. An enzyme block in one column will cause intermediate molecules to spill over into the adjacent columns, which perfectly visualizes why genetic enzyme deficiencies cause such complex physiological problems. Let us summarize the key takeaways from our lecture. First, steroidogenesis is the vital biochemical assembly line that transforms raw cholesterol into life-sustaining steroid hormones like cortisol, aldosterone, and sex steroids. Second, the entire process hinges on the initial conversion of cholesterol into pregnenolone, the universal precursor, by the desmolase enzyme. Third, the speed of this factory is strictly governed by enzyme kinetics, which we can model mathematically to understand production limits. Finally, the actual amount of hormone in your blood is a delicate steady-state balance between how fast it is produced and how fast it is cleared. Mastering these concepts gives you a profound understanding of human physiology and the delicate chemical balance that keeps us alive. Great job following along.